Welcome, my dear students. Today we are going to expose all of you with the fourth lecture of week 5. In the last few lecture, particularly in the last two lecture, we wanted to understand that how these residues can be converted into some useful resources. And in the last lecture, we categorically discussed about the conservation agriculture, particularly with a reference to residue retention mechanism. And there are multiple machineries we mentioned in that particular class. There we have seen that how these machineries are efficiently doing this particular operation in retaining those residues and making use of the best use of this particular residue. In today's class, we are just going to elaborate and we are just going to extend that particular portion basically with reference to your composting. Basically, whenever composting we coin this particular word composting, the thing which comes to our mind is it's a process, it's a very natural process which is mediated by mostly by your microbes, and we wanted to convert those residues into a nutrient enriched product, and which is like soil. And if we can do it very efficiently within minimum period of time, definitely we can reach to our goal. So in today's lecture, we are going to talk about these particular components of your composting. And once I am saying this particular composting, I mentioned that this is a wonderful and sustainable way to manage your crop residues and turn them into your valuable organic matter. And when I said organic matter, definitely I am talking about that enriched nutrient resources. And there are multiple objectivity or importance of this particular composting. Definitely it's a sustainable method for managing your agriculture residue, and we wanted to return back that particular nutrients, whether it is macronutrients, NPK, whether it is micronutrients, whether it is secondary nutrients, back into your soil and easily assimilable form. Once it is in easily assimilable form, definitely plant doesn't have much of stress to take those particular nutrients. And it also enhances your soil fertility, number of times I mentioned that, improves your soil structure and reduces dependence of chemical fertilizer, which is very, very important, because these chemical fertilizers are all energy intensive. So definitely it is going to boost the microbial activity, leading to a better nutrient uptake and recycling process, nutrient recycling. Effective integration of composting techniques can definitely and significantly reduce the dependency of those synthetic fertilizers. So that particular compromise, that how and what is that extent we need to compromise, that we have to decide, and it is based on the nature of residues from which we are going to make that sort of composting. So while promoting long-term agricultural sustainability, we wanted to make sure that our natural resource base, particularly with reference to soil, water, this microbiological diversity, remains intact and improves crop productivity. Finally, our objective is always we wanted to get the maximum out of these particular resources. And if it is that, definitely you are also ensuring your environmental component as well. That means we will be able to adapt multiple stresses within your environment, whether it is drought, toxicity or diseases. And there are key factors, particularly once we talk about this agriculture residue composting, the composition of substrate, the profile, the nutrient profile of that particular substrate, the pH, the structural and biochemical composition which I mentioned, nutrient content as well as the nutritive value. These are very, very important, particularly in making that particular final product which is going to be of our use. Temperature. Temperature is based on this particular temperature, the nature of reaction is going to be governed. So optimal conditions for microbial activity, we must ensure that. And moisture content, proper water balance for effective decomposition. Carbon nitrogen ratio, which is very, very important, particularly if it is high CN ratio, definitely initially we will have some issues, but once that carbon nitrogen ratio gets stabilized, things gets improved. So this is very, very important particularly, and in some cases it may so happen that if we started working with this high CN ratio, definitely we will have issues particularly with reference to nitrogen immobilization. So there will be some sort of nitrogen stress initially, but it is going to be improved in latter part of time. So to balance, cow dung or inorganic nitrogen sources should be mixed. Always it is a mandatory. If we wanted to take care of all of them, definitely we have to make that sort of mixture. And once I am talking about those composting, so there are multiple methods are available, particularly for composting your crop residue, whether you talk about aerobic composting, which is most frequently observed, that involves the decomposition of residues by microorganisms in the presence of oxygen, and requires a regular turning of the pile to maintain airflow, which is very important. So we need to look at multiple times within a week, or maybe multiple turning is needed, and produces very high quality compost quickly if it is properly managed. We have anaerobic composting, where you don't, in absence of oxygen, we just mediate that particular process of decomposition, and this decomposition process occurs in the absence of oxygen, suitable for cell systems like biodigester. Generators, biogas, mostly in this particular process, definitely it is going to generate your biogas as a byproduct, which can be used as a source of renewable energy. As well as very commonly in the present context, vermicomposting, which actually uses particularly your earthworms, is coming up heavily, because it has a tremendous capacity to break down those particular resources and convert into your organic matter, and results in a highly nutrient rich compost, maybe rich in your NPK. So the nitrogen content most specifically and the potassium content always can be enhanced, particularly based on that particular resources which you are going to use, known as worm casting, and ideal for small scale operation. There are multiple chronology of events and multiple customized level of those sort of vermicomposting is possible, and people have exhibited that sort of vermicomposting in multiple places. In situ composting, this is the crop residue where it is being generated. We can always do that sort of composting in that place actually. So crop residues are left on the field to decompose naturally, and reduces labor and transport cost. We need not bring it to some other places for any sort of intervention. The only thing is we need to take care of your moisture, the temperature, as well as if you wanted to give some sort of substrates, or maybe if it is some microbial consortia, many a time people they use it, and ultimately the conversion is always converted to, as I said, nutrient enriched, or whether it is nutrient enriched product or organic matter. Window composting, this is involving arranging of your crop residue in long or narrow piles, basically known as windows, for decomposition, and requires periodic turning for aeration, suitable for large scale composting operations. And trench or pit composting, this is another mode of, so this basically the residues are buried in trenches or pits to decompose and save space, and reduces those odor issues, because during the process of decomposition, and if it is being done mostly in anaerobic mode, definitely multiple organic substances are being produced, and which can create that sort of obnoxious odor. So we need to take care of all that particular part. And once we are talking about this composting, definitely there are multiple stages are involved, right? Whether starting from mesophilic stage, thermophilic stage, then there will be a stabilization stage. So you can see from this particular figure that particularly initial phase with temperature between 15 to 40 degrees Celsius, particularly if you look at this mesophilic, this particular range, here you can always see the temperature is going to be around 15 to 40 degrees Celsius. And bacteria, fungi, actinomyces, they all rapidly metabolize basically those carbon rich substances. When I am saying carbon rich substances, definitely those carbon bonds, right? Maybe those cellulose, hemicellulose, lignins, so they are being converted. Heat is generated due to your anaerobic metabolism, and temperature fluctuates as organic matter transformation progresses. The next stage is your thermophilic stage, and where the temperature increases or moves to around maybe 40 degrees Celsius or more. So thermophilic bacteria such as bacillus is always going to dominate over here, right? And carbon compounds break down, leading to a slight temperature drop before entering the next stage. In the first stage, what we have seen, that there is a heat is being generated, and this stage also, there will be, slight temperature drop with a, if you are going for the next stage. So definitely that's the temperature drop is going to help to move to that particular cooling phase. As I was mentioning, that cooling phase, that fungi decompose mostly, particularly in this stage, in this particular stage. So your fungi is of this particular, whenever we talk about this cooling phase, so definitely fungi is going to dominate over here, and fungi decompose complex organic residue like lignin and cellulose. And actinomycetes play also a very crucial role in forming humic compounds through condensation and decomposition. And finally, we have that maturation stage. And if we look at this maturation stage where, things are getting matured particularly, and if we look at the temperature requirement, because, so therefrom you will be able to understand that how things are moving. That final phase marked by a decline in your oxygen uptake, and temperature is below 25 degrees Celsius. So at the beginning, you have a temperature rise, and then it reaches to maximum, and then it's, slightly goes down, and ultimately it reaches to your 25 degree below 25 degrees Celsius. So macrofauna and soil organisms contribute to decomposition, and earthworms were very active in this particular stage, and organisms metabolize phytotoxic chemicals, improving compost quality and aiding to plant disease suppression. This is very important, because which are very, very important mechanism through which we can suppress those diseases, particularly in residues, because if that residue or if the preceding crop is having some issues with reference to disease, so definitely it is going to be the succeeding crop is going to be affected, but during this process of decomposition, that can be taken care of. So what are the key compost characteristics? What we have seen, that we need to have a lower pH and reduced CN ratio. Because as I said, that if you have a higher CN ratio, definitely initially you will have some sort of nitrogen deficiency, right? So, so that deficiency will create some sort of immobilization. So immobilization is going to, so if you, within that particular context, if you are going to grow crops, definitely the plants is going to suffer your nitrogen deficiency. Higher levels of plant available nitrate with lower ammonium, reduced compost order, and stable organic matter with retained calcitrant carbon compounds, which are very important, which is giving to improve the particularly with reference to, because once we are talking about those buffering properties of this particular the converted product, this is very, very important. The next mode is your anaerobic digestion. In the last case, we mostly emphasize on the level of oxygen, and oxygen was dominant in making that particular process to be, or converting those particular material to either it is carbon dioxide or water mostly, because finally we wanted to have those particular product in the form of either carbon dioxide or water. So in anaerobic digestion process, this is a biological method which breaks down your organic matter, and this particular organic matter definitely, as I said, that in the absence of oxygen, and primarily to produce biogas, whether it is methane or carbon dioxide, right? And this methane and carbon dioxide constituting almost 55 to 65% of your methane, and 35 to 45% your carbon dioxide, along with traces like your hydrogen sulfide will be there, hydrogen gas will be there, ammonia will be there, and digested, a nutrient rich material which is basically used as a fertilizer. It has a very high fertilizing value. And what is that particular breakdown process? If you can see this particular flow chart, there the things have been depicted very nicely, that we need to have those feedstock preparation. I am talking about those residues. Initially, once you talk about those organic matter or those residues, which is having your protein, cellulose, hemicellulose and lignin sort of thing. So those materials or agriculture waste or livestock manure are collected and prepared, and these may involve shredding or mixing to ensure uniformity. That has to be done. And then the most importantly, your hydrolysis. Once we do that, so definitely you need complex organic compounds like carbohydrates, protein and fats are broken down into simple soluble molecules such as sugar and amino acids, because this becomes maybe the precursor for the next group of reactions. And then the acidogenesis, this is very important one. The products of hydrolysis are further broken down by bacteria into volatile fatty acids, alcohol, hydrogen and carbon dioxide. And to do this, once you look at these acidogenesis, so there are fermentative bacteria which are very, very proactive in making this particular process of acidogenesis. And then followed by, once you have those acidogenesis, you have your acetogenesis. So this basically what happens over here, the volatile fatty acids are converted into your acetic acids along with hydrogen and carbon dioxide. So once it is being produced, and definitely one can see over here, mostly your syntrophic, mostly acetogens are involved in this process of converting this particular, once you talk about those particular, volatile fatty acids and alcohol, and is being converted into your acetic acids along with your hydrogen and carbon dioxide. And finally, we have those methanogenesis. So these methanogenic microorganisms, they basically convert this acetic acid, hydrogen, your carbon dioxide and methane and water. And this is the stage where biogas is primarily generated. Once we talk about those biogas, basically, once you have those methane and carbon dioxide, basically your process of biogas generation is being initiated. And then definitely once you have those particular biogas is being generated, we need to collect that. So the biogas is collected, often refined to increase your, this methane content, as well as, and it is used as a renewable energy, particularly for heating, for your electricity generation or fuel. So, and finally, as I was mentioning, apart from this biogas, the other part is those digest, right? So these digested is being used, particularly utilized as a fertilizer, and they have a very high fertilizing value, maybe containing more nitrogen, phosphorus, potassium content, and some of your micronutrient or traces of your secondary nutrient as well. So the remaining material digested is processed and often used as a natural fertilizer or soil conditioner, which can improve your physical and biochemical properties of soil. So these digest is having, it's a huge capacity to improve your soil physical, chemical, as well as, as I said, biological properties. At the same time, so within this particular anaerobic digestion, what is evident out of this, what we understand that, well, once we have those crop residues, initially we need to, undertake the process of hydrolysis, then followed by your acidogenesis, then acetogenesis, and finally methanogenesis. So once this methanogenesis is achieved, that means you have been able to produce from those organic resources or residues, because I said initially you have a high carbon containing compound, maybe you may have a cellulose, hemicellulose, lignin over here, and finally you wanted to produce either your methane or your carbon dioxide, right? So this is going to be used as a renewable energy, right? You can always use this particular as a renewable energy. At the same time, once you use this, you will have a digestate. So these digestate is going to be used particularly for as a fertilizer, and this has a huge amount of, as I said, NPK. So what are the key factors which actually influencing this particular anaerobic digestion efficiency? Initially, already I mentioned that pH is going to be very, very important, and this particular pH range in between 6 to 8 is, and if you have a deviation, this is going to inhibit particularly your bacterial activity due to your excessive volatile fatty acid or ammonia concentration or carbon dioxide or acetic acid accumulation, as well as the nutrient availability. The trace elements like nickel, iron, zinc and cobalt are essential for your microbial activity. So in that particular process, it has to be ensured that it is having a huge amount of those particular trace elements, so that that process getting, or the mediation of that process is very fast, and requirements vary based on your feedstock, reactor design and waste composition. From the very beginning, I am giving an emphasis on this particular part, that the nutrient profile of those particular your waste material is very, very important. Particularly this is going to impact the process, or whether it is going to be very fast or very slow, right? Temperature, this anaerobic digestion operates under your psychrophilic condition, means less than 20 degrees Celsius, or mesophilic and as well as thermophilic condition. But the yield of your energy is going to be always different. So if it is thermophilic this anaerobic digestion is always faster. Faster in a sense of producing or converting those resources, and you can see over here 15 to 25 days is needed. And if we compare it with your mesophilic range, which is, which will take around almost 3 times, 45 days, but requires more energy and risks of your ammonia inhibition is there. CN ratio, I was talking about that CN ratio, that if you have a high CN ratio, you have a chance of nitrate depletion initially. But if you have a low CN ratio, you can always do that, means for those microbial activity to, to proceed, definitely, that is very important. So many a time, those mixing of those organic waste, means having a high CN ratio, low CN ratio can, can be a workable solution and can help your optimize this particular ratio. Volatile fatty acid, this is going to include your acetic, propionic, butyric and, valeric acid, which are key intermediates during this methane production. And excess of this volatile fatty acid can inhibit this methanogenic bacteria and reducing your biogas yield. So we have to take care on this part as well. The final part is organic loading rate. That means once we take about the system of biological conversion capacity, this is basically, this is measuring that capacity, that how it is capable to, convert them. Overloading leads to low biogas yield and accumulation of inhibitory substance. So we need to be, take precaution during that loading of those organic resources and for converting those particular part. And this fermentation of your agriculture waste biomass for bioethanol production, this can be another way of, converting it for useful purposes. And for achieving it, we have, 3 ways of doing it. Either we can go ahead with your solid state fermentation, or we can go ahead with your liquid state fermentation or submerged fermentation. This is another, people also, put it under that particular terminology as well, and very high gravity fermentation. So in all these different modes where we are going to use, different substrates for producing or converting it to a bioethanol. So microorganisms, basically what they do, they convert those complex organic molecules into simple sugar, glucose, xylose or L-arabinose for bioethanol production. And 3 major industrial fermentation I already mentioned. So these are those particular processes and the energy which is being involved, particularly for converting those simple, sugar molecule, and finally, so from this glucose to finally to your ethanol. So you need this sort of mediation, and this mediation is definitely mediated by some enzyme, and definitely it is always going to yield your energy. So once I am talking about this 3 different modes of this fermentation, whether it is your solid state, liquid state and very high gravity fermentation, I was giving emphasis that yes, the substrate is always going to be different. So substrates for your solid, if you think about solid state fermentation, so definitely it is your solid material like your, maybe wheat bran, rice husk or any lignocellulosic your waste material. But if, if it is a liquid state, definitely the substrates can be your soluble, basically either it can be, xylan, pectin or mannan, or insoluble like the previous case, maybe bran, straw, or dust biomass, which dissolved in suspended water, right? But in case of when we look at the very high gravity fermentation, there we involve those fermenting substance with high sugar concentration. This is very important. And once you have a high sugar concentration, definitely it has a capability or capacity to produce your high ethanol production, right? High ethanol production, you can always do that. So this particular mode, basically as I was mentioning, that this substrate contains significantly higher sugar levels, whether it is your sugarcane juice or maybe molasses, and starchy materials like wheat and corn, compared to traditional fermentation methods. So in the first case, the solid process, basically they involves those substrate selection. I was talking about those substrates, basically those solid materials, and then microbial inoculation is needed. So microbial, when I'm talking about microbial, so definitely fungi is involved, and among those fungi you have your trichoderma and aspergillus, they are very, very predominant, and then bacteria, yeast are also involved, followed by your controlled environment that we need to take care, whether it is temperature, whether it is relative humidity, whether it is aeration, as well as the product extraction. That means whatever metabolites is being extracted, that has to be converted with more efficient energy material. But in case of liquid state fermentation, here almost similar. You can see over here the nutrient solution preparation, those essential nutrients are needed, And then your microbial inoculation, bacteria, yeast or fungi are involved. Aeration and agitation is needed particularly for this liquid state fermentation, where you need oxygen supply as well as those mixing. That means that agitation is, that physical agitation or mechanical agitation is also needed externally, as well as monitoring condition. It's related to, pH, temperature and those dissolved oxygen, right? So this is important, and product extraction like the previous case. And in the last case, where, as I was mentioning with reference to those substrates, that sugar concentration, material with high sugar concentration, microbial inoculants are definitely it's needed, and particularly we always prefer yeast, right, whether it is Saccharomyces cerevisiae, which is a predominant one, and stress management in optimization, particularly with reference to your temperature, pH and nutrient supplementation, as well as application. These are very, very important. And also there are possibilities that we can also produce bioelectricity generation from this crop residue, and this is also, I already described most of those methodologies, and basically it is based on this anaerobic digestion, where basically microorganisms break down those organic matter in the absence of oxygen, producing biogas. Gasification, crop residues are thermochemically converted into your syngas, which is basically a mixture of your methane and carbon monoxide and hydrogen, which powers turbine and engines. And microbial fuel cell, these microorganisms directly convert your organic matter into electricity into bioelectrical system. So these approach aligns with global efforts to reduce your reliance on your fossil fuels and mitigate climate change. So these, mode of converting this particular waste to your bioelectricity, because this is, as I said, this is the innovative one and sustainable approach, where we can, harness this renewable energy, and converting, we can always convert this agricultural waste, whether it is your any lignocellulosic crop residues or rice husk, wheat straw and sugarcane bagasse into your electricity. You can see over here that from this agricultural waste in particularly, whenever you talk about this microbial fuel cell, and this is being, the series connected with your microbial fuel cell, and ultimately your electricity is being generated. This is, something which is happening right now. In this way, one can see that, how, and in most of the cases, innovations are happening in this particular field. So in today's class, what we try to understand that, yes, apart from, because we started from the second class onwards, that how this waste can be converted into some useful resources. So in today's class, we have specifically given emphasis on composting. So composting, whether we talk about aerobic composting or whether you talk about those anaerobic digestion. So in that particular process, there are multiple, the conditions are being taken care of, and in that particular condition, whether you talk about pH, whether you talk about temperature, whether you talk about relative humidity, so everything, those conditions are being streamlined, and it is being monitored, because this is going to affect the efficiency of this particular production of your, whether you talk about compost or whether you talk about electricity, whether you talk about those production of your biogas, or whether you talk about the production of your those digested, right? Digested, which is a byproduct of that biogas productions. So these actually we have tried to understand in today's class. So thank you very much. We will continue this class with some more developments, particularly in this area, in the next class. Thank you very much.